So welcome to the National Oceanography Centre, Southampton. My name's Rachel Mills. I'm the head of Ocean and Earth Science, which is the university part of this building. And obviously we share the building with the National Oceanography Centre, some of whom are here today giving talks as well. So this is the Institute for Life Sciences conference, and it's called Life in a High Carbon World. And this is our first conference down on this site, I think. And obviously the Institute for Life Sciences is led by Peter Smith, who's sitting down the front, and we'd like to thank Peter for everything, and particularly Jeanette for all the organisation. So I've been very impressed so far. We're going to keep to time, and we're going to have a really exciting day. First of all, I want to thank you, people that have come from obviously this building, the rest of the university, from across the city, the region, and the wider UK. In particular, I'd like to thank our keynote speakers who've come from Bremerhaven, from Exeter, and from London to join us to stimulate these discussions. So thank you for coming. And I guess I wanted to start by saying something that I bet all the speakers say as well, but I get to say it first of all. Atmospheric carbon dioxide levels are rising and have risen to this year. They're consistently exceeding 400 ppm. And this is a really important milestone. It has really hit the press, it hits the public imagination and understanding about what our future might be. What made me think, actually, was this year we're celebrating our 20th anniversary of being in this building. So the Oceanography Centre has now had 20 years on this site. We're still here, and we're celebrating that this Friday. And it struck me that in 1995, when we moved into this building, carbon dioxide levels were only 360 ppm. And I remember teaching at that time, telling the students that this was a really high and increasing values of carbon dioxide. And every year I've had to change my slides and update that number and change the exam question that tests them on that as we've moved inexorably towards 400 ppm. In 1995, the IPCC second assessment was published, and that was really the first statement that the balance of evidence suggests a discernible human influence on Earth's climate. And I think what struck me was that by the fifth assessment that was published in 2013, we haven't actually moved on that much. We're still, we've reduced the uncertainty, we've put some numbers on the likelihood, we've made much more definitive statements about what's happening, but essentially we're still talking about the balance of evidence being that humans are influencing climate, and yet the rate of CO2 increase hasn't slowed down. So I think this is what really has stimulated today's conference. Carbon dioxide levels haven't been this high since the Pliocene. 3 or 4 million years ago was the last time we saw carbon dioxide levels this high on this planet. And while we have to use clever proxy measurements that we're going to hear about today to understand what the planet was like at that time, we do know that the temperatures were significantly higher. We know that they were particularly higher at the poles. We know that sea level was higher, the Arctic was ice-free, the surface ocean was more acidic. So we know the world was very different under 400 ppm carbon dioxide. And it's clear to us that our future is going to hold significant environmental change. But it's really hard to predict, and that's where you guys come in. It's really hard to predict the rate of change, the scale of change, and particularly the local impact of change to the south of England, the UK, Europe, and beyond. So the aim of today is to bring together scientists from lots of different disciplines from across the university and across the region, across the UK, to bring different perspectives to this, to stimulate new discussions and to start new collaborations. And I hope that's what we'll get out of today. I was going to say a little bit about the IfLS, for those of you that don't know what it does. I've already mentioned Peter Smith. But the IfLS exists to foster these sorts of cross-disciplinary links in life sciences and the adjacent disciplines. The Institute has just celebrated its 4th anniversary, and it's great to see global change emerging as one of the grand challenges. The global change grand challenge is led by Jörg Wiedemann, who's one of the conference organisers. You're going to see a lot of Jörg today. Along with Phyllis Lamb, Jessica Whiteside, and Chris Horton have organised today's meeting, and this programme of really exciting talks. So thank you to all of you. And I look forward to hearing new things from outside my discipline and to be part of the discussions about the future of this planet. Okay. So that reminds me, I've got a few more minutes, I'm just going to say a little bit about housekeeping. We've just instigated new swipe access to the building, those of you that haven't been here for a while. So to get anywhere in this building, you will need to use your card. And that includes the barriers that are just outside here. So if you want to go to the toilets, you'll need to, and you go out that door, you'll need to use your card to get through the barriers. And they work by placing the card on the little adjacent to the barrier. You have to actually touch the barrier with the card. Those of you from outside have all been given a card, and you'll have to keep it with you at all time. Okay, so that's the first thing. The second thing is there are no planned evacuations today at all. So if the alarm goes off, you will hear it. It is a continuous, very, very loud siren, and you will need to exit out of any of the fire exits and go out the front revolving door or any doors that you can find. And essentially, we need to we go to the front gate. So the muster station, if you like, for this part of the building is the front gate in the car park. So we'll to do that. Okay, so if you hear an alarm, do that. I've broken it, I'm afraid. And then the last thing I just wanted to say is, can all speakers please keep to time? We've got a really packed schedule and not much time to, in terms of between the talks. And we'd like to have some discussion at the end, so please, speakers, keep to the time allotted. And with that, I'll hand over to Jörg. Thank you. I think I've broken it.
So thank you very much, Rachel. Thank you for the introduction. Thank you, everybody, for coming. It's my great pleasure to introduce to you Professor Hans-Otto Pörtner. Hans-Otto Pörtner comes within a round trip over Europe, so he flew in from Bremerhaven, where he had a PhD examination yesterday, and he's moving on to Berlin before returning then to his home institution. So you might have heard his name in the context of thermal adaptation. That's where he produced a lot of seminal work, but he's also broadly interested in the effects of climate change, in global warming, ocean acidification, and he studies these effects at various levels, from the molecular level to the ecosystem function, using various techniques. And we will talk today about these impacts on ocean biology, physiological underpinnings, projections, and uncertainties. And this is his speciality, so we can be certainly excited to get some new insights. So he already started working in this area on ecophysiology when he did his PhD in Münster and the University of Düsseldorf. Afterwards, he moved on. He was a fellow of the prestigious Heisenberg scheme, where he spent some research time in Dalhousie University and at the Lovelace Medical Foundation in Albuquerque. So now he's head of the Department of Integrative Ecophysiology at the ABI in Bremerhaven. And apart from his research, he's also active in other fields. For example, he had a number of contributions to the IPCC reports, for example, as coordinating lead author and also as contributing author to many of his documents. And that is increasingly important, and I hope that we can also learn something from him in this direction today. So thank you very much for coming, and we're looking forward to your presentation. Thank you.
Thank you, Jörg, for your kind introduction, and thanks to all for the pleasure of being here and being able to talk to you about the research that we've been doing recently in the context of climate change impacts, mostly focusing on marine issues and marine animals. And I would like to start my talk with a reference to Article 2 of the UN Climate Convention, that climate change should not happen to an extent that it causes dangerous interferences with the human and the natural systems on this planet. We should allow ecosystems to adapt naturally. We can discuss what that really means, ensure that food production is not threatened, and enable economic development to proceed in a sustainable manner. And part of the work of the IPCC, together with the UNFCCC, is to find out where actually these thresholds should be set and the long-term global goals should be set with respect to how much climate change can we allow to happen. Now, focusing on the oceans, according to emission scenarios, the oceans are warming. Okay, oceans are warming, and we see different trajectories of ocean surface temperature here. And I'm contrasting in this talk two emission scenarios, which are now called representative concentration pathways. Essentially for us, it's important how much CO2 do we allow to accumulate in the atmosphere. And there is this business as usual scenario here, which brings quite a strong warming trend, and there is an ambitious emission reduction scenario at the bottom end. And as you can see, here's the phase of observations. We have already seen some warming in sea surface temperature. There is a phase where it really doesn't really matter which pathway we are going until close to mid-century, but then we are now making the decisions where temperatures will be developing after that. And the same is true for another trend in the oceans, which is the acidification trend. And here you see that with strong CO2 accumulation, we will see a stronger decrease in pH than with an ambitious emission scenario. We certainly have to consider what this means for ocean life. And then in addition to that, the oceans are losing oxygen. Here you see that there are already some historical observations of a decline in ocean overall oxygen content. And the projected trends also depend on the emission scenarios. And there are some, although the overall decline is only a couple of % over time, there are areas which are strongly expanding, those with the lowest oxygen concentrations, like oxygen minimum zones and the suboxic water layers. And certainly a question is, what does this mean for ocean biology? We have to start sort of with the question, what are we currently observing? And in the talk, you will always see some temperature indicator show up on the right-hand slide, not on all of them, but on many of them, indicating sort of what kind of temperature change I will be talking about. So we've already seen, and this is a global surface temperature indicated there. And we have already seen an about 0.8 degree warming of the world. As you can see here, there's some variability, but essentially it has affected to a large extent all continents and the oceans. And what we are observing is that organisms are moving, and they are moving to different degrees. So there's a change in the geography of life in the oceans that is driven by warming, and we know that the largest effect comes from the warming trend. So when we want to understand the effects of climate change, we have to try and understand the temperature biology of the organisms. We have to try and understand the thermal limits and the temperature ranges in which organisms actually like to live. And we have been thinking about this already quite early, and done a simple sort of comparison that prokaryotes, unicellular eukaryotes, unicellular algae, plants, and metazoa have different thermal limits and ranges, possibly associated with their organizational complexity. Because if we look at which temperatures are being reached and tolerated within these groups, we can see that the prokaryotes, bacteria and archaea, are going beyond 90 degrees, whereas the unicells among eukaryotes live up to 55 to 60, whereas then those more complex organisms that either have additional cellular organelles or plants and metazoa only live up into the low 40s. So that is the brick wall that these organisms will be hitting, and for the most complex organisms, this is getting close to the maximum temperatures that we actually observe in the oceans. And the question to be asked is really, what are the mechanisms, what are the explanations for this? So we were thinking about looking at the highest complexity levels, which would be membrane functions, molecular functions in the prokaryotes, compartmental coordination in the eukaryotes, which have more cellular organelles, and then organelles and tissues and central functions in the higher organisms. Okay, these were some nice ideas, but then we wanted for the IPCC report to put the numbers and check the numbers for those more consistently, asking the question whether limits are set at the highest levels of functional integration. So we did a meta-analysis, and this is a tool that is becoming more and more important with the number of and with the amount of information that becomes available in the climate change area. If you think about that, until I think the third or fourth assessment report, we had about 30,000 papers on climate change, and now we have about 30,000 papers on climate change per year, which tells you about the exponential increase in activities, in scientific activities in that area. So what we did, we were comparing the upper thermal limits in the different marine environments, comparing hydrothermal events to non-hydrothermal areas, very simple categorization, and we saw the trend that was already known. Multicellular eukarya had the lowest maximum heat limits, and the unicells a little bit higher, bacteria and archaea especially much higher thermal tolerances. And this would also relate to the width of the thermal ranges that these organisms would be living in, like the multicells here, the unicells a little wider, and the bacteria and the archaea actually the widest thermal ranges. If we put this sort of on a scale, we see this increase in thermal limits, and we were focusing on what we would consider the sublethal thermal range, because the lethal temperatures are really often beyond what the organism actually sees in its natural environment, and after all the constraints that it has already been living through, we see that actually the maximum ocean temperatures are getting close to some of the thermal limits, indicating what will be happening if those ocean areas will be exposed to continuous warming trends. We then thought about a simple complexity indicator, considering the numbers of compartments that these organisms would have to coordinate. And there is this simple relationship here. If we consider the temperature maximum for growth for these organisms and this complexity indicator, which essentially is this number of compartments evolved, we see such a relationship indicating that complexity actually would be involved in these relationships. Another issue to consider is, what does this mean in terms of where the organisms live? Not at all temperatures they do well, they do differently. What is the basic principle here? And one of the earliest principles that has been elaborated on in evolutionary biology is the one on thermal reaction norms, the thermal performance curves. And you see that these thermal performance curves, that each organism kind of lives in a limited range of temperatures, has a thermal optimum, it does where it does best, and then performance goes down beyond, goes down beyond and below that thermal optimum. And these thermal performance curves cover different temperature ranges. So we have to think about mechanisms that cause organisms to live in different temperature ranges, and what these mechanisms actually, and how these mechanisms change with the thermal limits, and how evolutionary adaptation sort of can, and on what time scale actually, which is a pressing question, on what time scale these changes can actually occur. So with this kind of background wisdom, we went into preparing the chapter, the new chapter on ocean impacts of climate change on ocean biology in the 5th assessment report. And you see here the thermal performance curve. You see the specification for animals, as we think that the highest level of functional coordination there actually involves the cardiocirculatory system, the oxygen supply capacity, the relationship between oxygen supply and demand, on top of all the other processes that are being affected by temperature changes down to the subcellular and molecular level. So if this is sort of the highest level of integration, then we see that there's indicated there is some dynamics in that temperature change, and that involves not only temperature, but CO2 and hypoxia we know, and there would be a natural link here. would influence the dynamics of the thermal performance curve. And this performance curve would then relate to the temperature-dependent biogeography of the species. I think that is pretty firm knowledge, which it wasn't 10, 15 years ago. It's now firm knowledge, appreciated that in the oceans the biogeography is largely temperature dependent, with some influence of the other factors. And that is still a tricky area of research to be investigated. So we see some compression here indicated in habitat range on the southern, if we go for the northern hemisphere, on the southern side, and expansion of the biogeographical area in the north, and the northward shift of the abundance of a species. This means, if we consider these principles, that species, if they cannot, well, first of all, this involves, if it involves all species, it also involves fishes, and fishes have an economic interest especially, and that's why here the borders between country A and B are indicated. If fish stocks move across those borders due to temperature, then certainly this will cause some changes in human interest and maybe some political complications. So here in the south we see local extinctions, in the north we see invasions. At the same time, if they cannot fully follow the temperature drift, there will be a reduction in body size. That is also one of the overarching principles to be seen in the oceans. If we look until mid-century, we would be expecting a shift in distribution to cooler waters in about something like 30, 40, 50 kilometers per decade, depending on the species. And also fish would move to deeper water to stay in the preferred temperature range. Now, certainly we were interested to understand more about what is the physiological background. And if we sort of have an understanding of these principles, we have to calibrate it against what's happening at ecosystem level. So we were bringing ecological, physiological information together for, in this case, a species living in the southern North Sea, the Ilpaozoarchus viviparus, comparing the change in abundance with temperature. In extreme summers, you would see a decline in abundance. What you would also see that the largest individuals would disappear first, which would be in line with expectations. Here you see the temperature-dependent growth curve, which goes down, sort of in the same temperature area. And you see here how the blood flow changes run into a constraint beyond a certain temperature threshold. And then at very extreme temperatures, you see oxygen deficit developing at systemic and at tissue and cellular level, indicated by an accumulation of anaerobic metabolites. But what I want to indicate is that the first constraint is actually at a temperature which is not yet lethal. But it's a reduction in performance which is constraining the well-being of the organism in a system. And this is, we call this the oxygen incapacity limited thermal tolerance hypothesis as a physiological principle that underlines the thermal limitation of an organism at ecosystem level. We did some of these investigations with our NMR laboratory, where you see here the bright spots indicating the blood flow in the major vessels, blood vessels, under these conditions. So there's some background information that needs to be considered when we look at those data. I put the growth data in. Growth you would measure about on extended time periods. This involves thermal acclimation, and it involves energy budget trade-offs. So organisms invest the energy into growth, and if they don't do any longer, the energy goes somewhere else. May go into maintenance, may go into other activities. At its southern distribution limits, a population lives at the limits of acclimation capacity, and then once it goes beyond the optimum and goes beyond what we call Peyer's limits, we see a decline in performance, which then leads in a decline in field abundance. And limited growth relates to limited cardiocirculatory capacity in relation to demand. So these were kind of data in a sessile fish species, indicating that this is a principle that may be operating. We have collected data from representative species in many animal phyla, and I cannot go into showing you all these details. But here I would like to emphasize another example from a Canadian group that was working on this in parallel, but in this case on an active fish species, a swimming fish species. The sockeye salmon migrating into Fraser River and reaching into different river systems depending on where the spawning location is. And what you see here is that the temperature profile of the river system matches sort of the temperature profile again of the performance curve of that respective population. So there is some evolutionary adaptation to the specific sites where the population spawns. And now what they observed is once the river gets too warm, performance declines, migration stops, and there is a huge increase in mortality. So these are fish that are active, that are continuously active during a certain life phase, and this is steady state performance. All of these terms are important to emphasize because there is some misunderstanding currently developing in the literature about what kind of thermal limitation we should be looking at in this context. And here we have a recent example, again a different group of animals, in this case is crustaceans. Actually the first data we got in this context was in crustaceans, and here you see how oxygen consumption changes and ventilation changes with temperature in 3 different amphipod species. You may know that the group of amphipods reaches a large diversity in Lake Baikal, which has the largest sort of freshwater body on the planet. It has 20% of the freshwater resources, which is I think an impressive number. Anyway, so if you see those temperature-dependent changes in oxygen consumption, again, trying to identify these thermal limits that we are working with. Here's the Peyer's limit, the critical limit, which is the one indicating the transition to anaerobic metabolism and the functional constraints. What you see is that at critical limits, mortality rises. And actually, there is one observation, and you sort of see sort of differences between species, but the same kind of principles, critical temperatures, onset of mortality, ventilation being involved in the thermal limitation. But a critical observation here is that this species is migrating to deeper waters, not at critical limits, but again, once it reaches Peyer's limits and beyond. So a performance decline, sort of a sublethal constraint on performance, is causing an ecological response. So which brings us to think about, okay, what do we know and what can we say about the thermal range that the species is living in? And here I put quite a bit of information in. Just to walk you briefly through, we have here the Peyer's limits, upper and lower, and then the critical limits, the transition to anaerobiosis, before, and this is all important before actually we reach the lethal temperature and the animal just dies. And acclimation can shift those limits. We know that the oxygen supply, a demand relationship, and the resulting oxygen levels in body fluids plays a role at the tissue level. What I'm indicating here is the performance curve being placed into this physiological framework. And the limits here indicated between lethal limits would be what, according to traditional ecological understanding, would be the fundamental niche. And there are factors interacting, oxygen, CO2, temperature, shaping the performance curve which relates to growth, reproduction, development, foraging behaviors, immune capacity, all these things an organism needs to be doing at a certain time in its natural environment. How do we go from there to the realized niche, which is more important at ecosystem level? What is things are happening beyond the Peyer's temperature, but not and prior to the critical temperature? What is causing effect to set in at ecosystem level? And there was one activity recently that really helps us to address this question. If we look at the metabolic rate of a species in relation to temperature and what it does over time, we can distinguish different levels of metabolism. One is the resting metabolic rate, and then the sustained metabolic rate in the field usually is higher than that, because all of these performances need a certain level of energy. The resting metabolic rate would relate to physiological survival and the fundamental niche, and this, what we call the routine metabolic rate, would relate to ecological survival and may relate to the realized niche. And there is a certain ratio of field to resting metabolic rates that comparative physiology has compiled over time without really relating that to biogeographical information. There is a ratio between 1.5 to 5 in birds, mammals, lizards. We have a certain ratio as well. And you wonder how much does this ratio vary across the biogeographical range of a species. So we joined forces with some modelers, with Curtis Deutsch from the University of Washington, and looked at this recently, how temperature, oxygen in the oceans relate to this routine metabolic rate. And we did this for one of the species we have been working on, Atlantic cod. And this is sort of the indicating the limiting level of routine metabolic rate, and you see how nicely this correlates sort of with the biogeographical limits, southern biogeographical limits for that species. So there seems to be a critical metabolic index, sort of which, if we relate that to the performance window, narrows the actual performance that is possible and needs to happen at ecosystem level. And we can further resolve for this. If we look on the Western population, it actually undergoes some migration between seasons, going from 41 north in the fall to 37 north in the spring. And we see that this migration also relates to this critical metabolic indicator. Whereas on the eastern side of the Atlantic, we see some movement across water layers. So, and again, this relates to the critical metabolic index. So what we can say, if we take this performance window concept and the related underlying principles and take it sort of into ecosystem, the window becomes even narrower. And here you see this performance window again indicated here with the threshold, and there seems to be sort of a limiting level of performance that the organism needs to undergo at ecosystem level, which we would call routine metabolic scope as a link to the realized niche at ecosystem level. This has all sorts of implications, because if we sort of take this further, we will understand how this kind of performance is being shaped, what are the interactions between mechanisms and the scope and limits of the realized niche. And this brings laboratory people and ecological people together. And also it will bring us to understand how species interact at ecosystem level, because they can interact there where their thermal ranges overlap. And if there's a dynamic in the thermal ranges, there's a dynamic in coexistence ranges and the related ecosystem level consequences. But this is kind of, again, sort of reaching into the future, because these changing species interactions based on physiological principles haven't really been investigated and are certainly not yet considered in any modeling exercises. But what we can say is that wherever we've looked for a certain life stage of a species that's important, whether it's the adult or a larvae or whatever, there may be critical life phases where the OCLTT principle comes into play. And I've just listed these examples here as we know about it, considering that the thermal window changes its width and position on the temperature scale throughout the life history of a species. There are spawners where we know that they undergo thermal constraints like Pacific salmon and spawning migrations. There's a sensitivity of Atlantic cod to warming winters, which also affects the adult spawners. Then we know that the growing adults go and run into progressive thermal constraints. The window narrows down once animals grow up, where that's the example of the eel pout in the Wadden Sea. This is setting the southern distribution boundaries of Atlantic cod. Baikal amphipods then move to deeper waters, and you see the same effect in the literature for Atlantic salmon. Also, there are examples on where oxygen limitation has been shown in the water fleas abundance in relation to seasonal temperatures. Interactions, there is some indication that this really works. Thermal ranges of competing sardines and anchovies are affected. This is an example, and the larval stages certainly are important here too. And a lot of more work needs to be done considering these relationships. Now, during the final section of my talk, I would like to go back to the initial question. What kind of long-term global goals do natural systems suggest that we should be going for? And this is something very important because many human systems don't give us this specific information. And the natural systems can really provide the guidelines for that. Let's start with the vulnerable ecosystems that, according to the evidence reported in the IPCC assessments, are currently under pressure. And one of them is the Arctic summer sea ice system, which, according to observations, is on a decline in summers, and according to different emission scenarios, will reach different levels. If we go business as usual, the trajectory that we are currently on, we will have a summer ice-free Arctic by mid of the century. Some people expect that already in the 30s. You see there's quite a range of uncertainty there. This again relates to the temperature here. And there's another vulnerable ecosystem, which are the warm water coral reefs, which are under combined pressures. And one of them is that these healthy corals here, if they are exposed to too high temperatures, they lose their endosymbionts, they bleach. And if temperature doesn't go back soon after that, this reef may turn into the desert because corals do not survive the resulting energy limitation from the loss of these symbionts. And that the reefs currently are under pressure already in the present world. There is this example from the Great Barrier Reef, which has since the 80s already lost 50% of its life coral cover. And if we talk about the reasons for that, they are various. So it's not always climate change, which makes the attribution of changes to climate change often quite complicated. There is a predatory sea star working on those reefs. There is enhanced exposure to extreme events, and there is this bleaching that also contributes to the demise of the coral reef, which you can see here is probably and will be continuing into the future. So what are the projections then if we go into the future? And what do they tell us about the long-term global goals? Let's talk about ocean productivity very briefly. We see here that productivity will be shifting, it will be moving towards higher latitudes, and there is sort of a decline at low latitudes, a so-called desertification of warm water areas in the ocean. And if we go into the sort of business as usual scenarios, this trend is being exacerbated. Productivity largely shifts into the high latitude areas. And the projections tell us in a 2-degree world by mid-century that higher organisms will follow. And this kind of translates this into fisheries result. We see a reduction in biodiversity and ocean productivity in low latitudes in all those places where developing countries have their artisanal fisheries. They will suffer from these trends, whereas we will see an increase in productivity, especially in the north. In the south, the krill will be affected. That is what these red colors are about. So large-scale shifts in fisheries will be happening under climate change, and many developing countries will run into problems to adapt to these situations because they have a small human adaptation capacity. This combines with the already existing pressure of overfishing. If we look what the present situation is, there is a decline in predatory fish biomass, those fish that we like to eat, already by 70%. For comparison, I'm adding information from the terrestrial side because people are also discussing how does climate change affect crop productivity. And what we see here is that beyond 1.5 degrees, we are running into a high risk of severe impacts after 2050. So in a way, the constraints on food security on land and in the oceans go hand in hand in a world that is warmer than 1.5 degrees Celsius. And on top of that, in the oceans, we see a decline in oxygen, as already talked about, that is affecting ocean areas, especially the northern Pacific. It's also affecting the Atlantic, as the blue color in this case indicates the decline in oxygen content to be expected. And remember, I emphasized the relationship between temperature and oxygen supply and demand. And if we take this further, this will exacerbate the trend of removing the larger fauna in certain areas and pushing species from large to small. So there is not only a biogeography shift, reduction of body size due to temperature, but there is also a projected decline of body size and abundance due to the reduction in oxygen content. We will see a compression and loss of habitat in the oceans. And we have done this as part of the modeling exercise, where you see that in those areas where the oxygen content, like here the North Pacific, is already relatively low and will be declining further, and in the mid sort of low latitude areas here, and also in parts of the Atlantic, this decline in oxygen will, due to its effect on the critical metabolic index, cause a further reduction in available habitat to species under a business as usual scenario. So this should be a red light in a way for human society, because this is constraining distribution, body size, and also has an effect on diversity. And I've not talked about ocean acidification yet. Ocean acidification that has been reported to have begun in industrial times is presently going on with the increase in atmospheric oxygen attention. What we know is that ocean acidification affects the calcifiers in the ocean. And there are quite a few of them. These are the warm water corals. These are the green spots here. These are the cold water corals, which are the red spots here that have only been discovered in the 90s. And what you see here is that these different groups, and they are the other calcifiers that are being connected, especially in the continental margins, and have a high commercial importance here, indicated by the yellowish color. And what you see here is that these groups differ with respect to their sensitivity to CO2. There is an early onset in mollusks, there is an early onset in warm water corals. Crustaceans are less sensitive, and cold water corals are less sensitive than the warm water side. So depending on the CO2 ranges that we reach, we will have certain risk levels for this group. And these risks will be enhanced by warming extremes. And this is once again indicating what will be happening in a world that goes beyond the 2 degrees. Assessing risks is actually a very important issue in climate change, especially in the working group 2. We have used a risk concept that considered exposure, that considered the hazards first of all, and then the exposure to hazards and the vulnerability of the systems that we were looking at. And all of this would combine to a certain risk level. Climate change will amplify existing risks and create new risks for natural and human systems. That is one of the strong messages. Risks are unevenly distributed and are generally greater for disadvantaged people and communities. You've seen some examples from the oceans. And increasing magnitudes of warming increase the likelihood of severe, pervasive, and irreversible impacts for people, species, and ecosystems. And coming back to the question, where should we heading for? This is showing a global perspective of risk in the so-called burning ember diagram, calibrated according to the colors we see. And you see for some unique and threatened systems that already with an beyond 1.5 degree warming, and this is the reference scale here, pre-industrial, we will have a transition to high risk. And how can we once again illustrate that? Comparing risk levels at different global temperatures. That's where we are. That's where we possibly should be headed. That's the value that we do not want to exceed, and this is sort of where we are headed with business as usual. What are the key risks of impacts? What are the avoided impacts? Thanks. Now let's talk about what we have already talked about for the oceans in an 0.8 degree world. What are we currently observing? Species are being displaced. Some unique systems, coral reefs, summer sea ice systems, are losing resilience and spatial cover. There are some groups, like in mollusks, the pteropods, foraminifera, and bivalves, where we already see impacts of ocean acidification. Risks are still moderate, but may rise as climate change combines with other pressures. If we do such a risk assessment in more detail for the ocean acidification side, you see the burning ember diagram here, where according to the CO2 concentrations reached in the atmosphere, the risk is changing, and we are at 400 ppm presently. And you see we are close to a risk threshold. We're getting closer to a risk threshold if we just look at ocean acidification itself from yellow to red. If we combine acidification with warming, where we know the thermal ranges is narrowing for some species, and we may have a transition to higher risk, we see that actually in a 2 degree world, we would already be in a high risk area. And only the 1.5 degree world would actually define a world where the risk level would sort of be close to transition from moderate to high. This is the kind of information that natural systems can give us about where we should be headed with respect to allowing a certain degree of climate change. Let's talk about coral reefs. And here the situation looks pretty grim already, according to modeling projections considering the different emission scenarios. What we can say is that there is a trend for losing those coral reefs in a large percent coverage of spatial area, and that's the key thing. That means we will lose in large parts of the world the ecosystem services provided by those reefs, ranging from fishery supplies to coastal protection. And these modeling exercises indicate that global mean temperature would have to be limited to even 1.5, 1.2 degrees with an uncertainty range in order not to lose more than 50% of the coral reefs. And this is not yet taking ocean acidification effects into account. A very obvious thing is sea level rise. And if we look at earlier IPCC reports and even at the 2013 report, for some of these figures you think, oh, climate change is going to stop in 2100 because that's when all the figures stop. And that is due to the uncertainty involved with the modeling. And in the synthesis report, we have pushed this a little further until 2500. Business as usual scenario, atmospheric CO2 levels will lead to compared to an emission reduction, will lead to very different atmospheric CO2 levels in the end. Global temperatures will be very different. And what you see here is sea level rise with a huge range of uncertainty of where we might be headed depending on scenario. But if we want to stay on the safe side and keep sea level rise sort of at about 1 meters, which we would be headed by the end of the century, going for a 1.5 degree warmer world would be a wise thing to do, because as we have already heard, paleo observations as a reference during the last interglacial, when temperatures were actually 0.7 to 2 degrees warmer than pre-industrial, we had 5 to 9 meters higher sea level. And the last time when the atmosphere had 400 ppm in the Pliocene 3 to 5 million years ago, sea level was also in that range. So there are things to consider in the discussions, in the decision process, and importantly, to make decisions under uncertainty seems to be very important. Because, and this is emphasized by the respective risk assessment that is in the synthesis report, if we go for a low CO2 emission scenario and for a 1.5 degree warmer world, we will keep risk level at relatively moderate levels, but we will already have a high adaptation pressure on human systems, because many of those systems are located in coastal areas. And this is one point that we need to emphasize after looking again at the summer sea ice in the Arctic. Here again, there is a chance that if we go for a 1.5 degree warmer world, we may keep some of the summer ice in the Arctic. Okay, what I will be just have a couple of slides that kind of summarize what I've been talking about in terms of what are the avoided impacts if we go for the 1.5 degree world. Climate change velocity would be slow enough for most, including terrestrial and freshwater organisms, to follow. That is an aspect I haven't really been going into. Up to half of the coral reefs may remain intact. Sea level rise may remain below 1 meter. Some Arctic summer sea ice may remain, and ocean acidification impacts remain at moderate levels. But we already will see impacts like the capacity to increase food production to feed a growing population, is reduced with some scope for adaptation. Some unique systems are at high risk. More than half of the coral reefs may be lost, which is kind of the glass is half empty. Risks of combined ocean acidification and warming become more prominent. So this is a world which has sort of some severe impacts, but still has some positive messages involved that might be worth going for. And in a recent assessment where we summarized the IPCC insights with sort of an updated it for a recent conference in Paris, we were kind of comparing the scenarios again, the business as usual with the low CO2 emission. And you see the risk levels for the different groups, warm water corals, and for the bivalves according to ocean acidification effects here being red. But what you see is that these risk levels are purple for many of those groups. And what we also can say is that any adaptation measure in the oceans that we do, open ocean, carbon uptake, coastal protection, coral reef recreation, bivalve fisheries, aquaculture protection, finfish fisheries protection, and so forth, the adaptation measures we would be able to do in terms of mitigation, adaptation, protection, and repair have a certain scope in themselves. And this scope is becoming much reduced if we go down the business as usual trajectory. We just will be surpassing adaptation capacity. And that is a message that is important because adaptation is one of the main pillars of human adaptation, is one of the main pillars of present discussions about how to deal with climate change. Adaptation doesn't work without mitigation. Thank you very much.